Chapter 10, Section 2, Part 2. We're going to start here with looking at how antibiotic treatment might be a little different when you are talking about biofilms. Remember that biofilms are very difficult to treat. They are very different than bacteria that are free-living. Alright, so just individual bacteria that might be growing on a surface or in the tissue, in your blood, in your tonsils, that is causing you to become sick. When the bacteria get into a biofilm and the biofilm develops and is organized, then the bacteria do different things. They turn on different cellular signals, they act different, they talk to each other differently. And so it's not surprising that the treatment for bacteria in a biofilm is actually very different. And the reason for that is it's hard for an antibiotic to actually penetrate the biofilm matrix material. If you remember, I may have mentioned that a biofilm, the surface of the biofilm kind of acts like an umbrella. And if you're out in the rain using an umbrella, the rain hits the top of the umbrella and washes right off, and you are kept dry. Alright, in this example, the umbrella is the outer layer of that biofilm, and the rain would be the antibiotics. The antibiotics come down onto the surface of that biofilm, and it just kind of runs off, and the bacteria underneath the biofilm, inside that biofilm, aren't affected by the antibiotic because it just simply doesn't get in. And because these bacteria do different things, they look a little different, they act a little different, then the antibiotic treatment might also be affected in terms of their sensitivity. So some strategies to treating biofilm infections is to add medication or different chemicals that might interrupt the quorum sensing signals. Those are the different way the bacteria talk to each other when they're in the biofilm. You can also add DNase to antibiotics. That's going to help degrade the biofilm material, allowing the antibiotics to penetrate better. And then you can also impregnate devices with antibiotics. So biofilms in the body are very common on joint replacement material, on heart valve replacement material. And so as those are being made and synthesized, part of that process can involve being coated with antibiotics so that when they are placed in the body and a bacteria cell comes in and sits on that surface, the antibiotic is already there and it can kill off the bacteria before the biofilm actually develops. Now, looking at eukaryotic cells. Alright, this is a major issue, alright, because drugs designed to act on bacteria are ineffective against fungi. And if you recall from maybe case study number 1, where the person had developed fungal pneumonia and he was given antibiotics and the antibiotics didn't seem to be doing anything. That's because it was fungal pneumonia, and antibiotics don't work against fungi because they're eukaryotic. And so the issue is that fungal cells and human cells are very closely related. And so if a drug is going to be toxic to the fungal cells, it can also harm human cells. So there's actually very few drugs, only a handful of drugs that are antifungal that have been developed that are not toxic to human cells. Another drug or another issue is protozoans. All right, those organisms that cause infection in humans that are of protozoan or protist nature. There's even fewer drugs than fungal drugs to treat those kind of infections. Thankfully, within the United States, those infections aren't prevalent, but around the world, in tropical areas, they're still a major issue. And with some of these drugs, there's actually a lifetime dose that you can have because of the toxicity. All right, because the toxicity or the drug gets into the body, our liver and kidneys have to try to clear that medication by breaking it down. And so the drug is naturally going to concentrate, if you will, in the liver and kidneys. And so that can be very damaging to those tissues. So a person can take some of those drugs, but it's going to be in their permanent medical file, so to speak. And once they take that drug, then they can no longer take any more. And that is also similar to chemotherapy drugs. All right, now, action of antimicrobial drugs. All right, on the left side, you have the 3 modes of action: inhibition of virus entry, inhibition of nucleic acid synthesis, inhibition of viral assembly and release. So, you can't alter viral metabolism because viruses do not have metabolism. Right? They get into the host cell and hijack the machinery of the host cell to do their work, but you can't stop the metabolism of the host cell because it's not going to be specific. So you have to try to find some other avenue of drug activity to inhibit viruses. So if you can inhibit the virus from getting into the host cell, then it's just free-floating and it's not going to do anything. And the biggest example there is going to be influenza virus. And with the influenza virus drugs that act to inhibit virus entry, you actually have to start taking this drug within 24 to 48 hours of showing symptoms. Otherwise, the virus has already gotten into enough cells that the medication isn't going to help. Inhibition of nucleic acid synthesis. There's a variety of different drugs that can be used for this. One of the big ones is herpes virus infections. And then inhibition of viral assembly and release. One of the big ones is the HIV, the protease inhibitors. And so it's going to, inactivate the HIV protease. And so then inactivate the HIV virus. Alright, so that is the end of section 10.2. And make sure you go through your chapter objectives and look at these and start working on creating your study guide.